Hi, my name is Jacob McPhail. I'm a scientist at Stressmark Biosciences. We're located in Victoria, British Columbia, Canada, and we are a bioreagents company. And I'm going to tell you today about novel tools for drug discovery in neurodegenerative diseases that we've been developing for the past several years now. Now, Alzheimer's and Parkinson's diseases are the most common neurodegenerative disorders. Alzheimer's disease is the sixth leading cause of death in the US, likely closer to third in those aged 65 plus. So this is a massive problem, and despite significant impact and massive investment in research, a lot of clinical trials, no definitive cures are known. These diseases are both characterized by the presence of plaques in the brain, and these consist of protein aggregates, which include key proteins alpha-synuclein, particularly in Parkinson's, and amyloid beta and tau in Alzheimer's disease. Now, alpha-synuclein, tau, and amyloid beta share a common pathological mechanism, whereas these relatively disordered monomers form these toxic oligomers and these really hydrophobic fibrils that deposit in these plaques in the brains of these patients. And here at Stressmark, our goal is to produce active pathology-inducing aggregates, such as these oligomers and fibrils, in vitro for use as reagents for disease model development to really accelerate Alzheimer's disease, Parkinson's disease drug discovery. Now, starting with Parkinson's disease, it's characterized by alpha-synuclein aggregation. So again, we have this normal native alpha-synuclein, fairly disordered in solution. It does form some order when it's on membranes, but through this pathological process forms oligomers, fibrils, and these fibrils deposit in what are called Lewy bodies. And there's also significant evidence for transmission between different sort of infected, deposited plaque neurons to one another. And an important part of this is the presence of this phosphorylated serine 129 on these fibrils. So as these are deposited, they become more and more phosphorylated at this 129 position of alpha-synuclein, and that's actually a hallmark in Lewy body aggregates for Parkinson's disease. And it's going to be important because it's a way we demonstrate pathology within these disease models from some of our recombinant materials we use to induce Parkinson's-like disease. So we've generated several types of alpha-synuclein preformed fibrils in vitro. So here you can see the fairly pure protein on the left and these fibrils we've generated under TEM in the middle panel. And we can actually track the formation of these fibrils in vitro doing a THT binding assay. So as this THT molecule binds to these more hydrophobic amyloid fibrils, it actually shifts its fluorescent spectra. And basically, as you get more and more fibrils forming, you get more and more THT binding, and you get an increase in fluorescent signal. Now, it's important to note these reagents, we've expressed this alpha-synuclein in E. coli and then fibrillized it in a test tube, basically. So there's no phosphorylation present because E. coli can't post-translationally modify these with phosphorylation. So going into the experiments, there's none of that phosphoserine 129, and that'll be something we're looking for to represent a Parkinson's disease model. And we've been able to do this preformed fibril in both human, mouse, and rat variants of alpha-synuclein, and all of them seed monomers into fibrils in vitro. So in the first model here, we have these alpha-synuclein preformed fibrils on neuroblastoma cells. So these were initially isolated from a cancer, and they're commonly used to study neurodegenerative diseases. They're neuron-like cells. And what you'll notice, especially in the middle two panels here, at time zero, our phosphoserine 129, it appears to be a bit of a signal. So this is using antibodies staining these fixed cells at time zero here. And you'll see there's a little bit of phosphoserine 129 signal. That's actually just, we've run some controls, and that's just due to the binding of our secondary antibody, which is the fluorescent one, to the ER region. So that signal really is not there. After 3 hours, you can see a robust increase in this phosphoserine 129 signal. So that has to be occurring within the cells, as the fibrils we're adding don't have a phosphorylated serine 129. So we're inducing that pathological aspect of the disease. Here are 2F11 antibody, so that's an antibody we have that's anti-aggregate, so it will bind to these fibrils. You'll notice that also has a robust response, even within 3 hours after addition of these PFFs to these neuroblastoma cells, we see a massive increase in aggregates. So we're both recreating the phosphoserine 129 pathology and inducing aggregation of the endogenous fibrils here within 3 hours. So very quick, robust model of Parkinson's disease in cell culture. Now, we also wanted to demonstrate the actual transfer of these preformed fibrils between different neurons. And the experiment we designed here was a fluorescently labeled, so this BF555 fluorescent tag on these preformed fibrils. We incubated them with GFP expressing neuroblastoma cells. So they've been differentiated for 8 days. They're glowing green, they're expressing GFP, and we're adding the fibrils to them. After 48 hours of incubation of these, we then wash away the free fibril and then trypsinize these. So any floating around free fibril is going to be gone. It's only going to be the internalized fibrils remaining in these neuroblastoma cells. And then these are added onto 8-day differentiated iPSCs. So these are differentiated to form neurons. These are human iPSCs. And we're going to be looking at purple color here for tubulin. And essentially what we see here in this image is after 14 days, this sort of co-culture is imaged. And what you'll see in green, these are the GFP expressing neuroblastoma cells. You'll see some red in there that is the actual tagged fibrils. But what you'll also see is when you look at the tubulin-rich iPSCs, you'll see that the red fibrils have actually transferred into these iPSC cells. So evidence of direct transfer from these neuroblastoma inoculated cells into these iPSC cells, which is pretty cool to see this transmission. Then we have actual mouse models using these PFFs. So here we're looking at phosphoserine 129 staining in a mouse brain after 30 days. So a single unilateral injection to the dorsal striatum. And you'll see on the top here, looking at monomer, there's really no staining at the 2 concentrations tested. When you look at the preformed fibrils at both concentrations, you will see that darker staining, and especially at the higher 5 microgram injection, you actually see it transmitting to the other hemisphere of the brain, which is pretty exciting. So these PFFs have proven functionality in generating Parkinson's disease models, both in neuroblastoma cells and iPSCs, as well as actual mouse models. And just a reminder, so that's the PFF side of life. The oligomer species, there's evidence out there that this is actually the more toxic species, and this is what should be targeted therapeutically. So we also want to be able to recreate those. And for alpha-synuclein, we've been able to generate these kinetically stable oligomers in vitro. So you can see here on the left panel, this is a native gel. Normally, alpha-synuclein on native gel runs between 75 and 50 kilodaltons, and you'll see it runs above 250 kilodaltons there. In the middle, we have an SEC trace, and we've actually been able to freeze thaw these and incubate them for 37 degrees for up to 14 days we've tested, and they don't actually lose their oligomeric structure. So you can see that there's a small amount of monomer within the prep, but the majority of it is oligomer and stays oligomer and elutes the size we would expect of an oligomer. And on the right, that is, so these globular structures rather than these longer fibrillar forms for oligomers. And the important thing to note here is these are generated in a native state, so there's no inducer inhibitor, there's no dopamine, there's no other materials added to force them into this. It's just them alone in an oligomeric state. And it's important to note too that these oligomers are structurally different than the fibrils. They're not just little pieces of fibril that have broken off. The actual secondary structure of these is quite different, and they are all also soluble. So this is far UVCD, and basically it gives you an idea of the relative amount of alpha helical beta sheet disordered structure. And what you'll notice here is the oligomers tend to be much more alpha helical, whereas the fibrils, of course, are higher beta sheet, that amyloid type structure that the TH2 binds to that we track during those seeding assays. So there are fundamental differences in the structure of these oligomers and fibrils. They're not just pieces of each other, they're distinct. So we then took these and tested them against primary rat dopaminergic neurons. So here we're looking at our control and our treated primary rat neurons, and we're staining with MAP2 for dopaminergic neurons, and we're also staining for phosphoserine 129. So MAP2 is in red, phosphoserine 129 is in green. And so you'll see anywhere there's yellow, which is indicated in arrows just for clarity, overlap between dopaminergic neurons and phosphoserine 129. So these non-phosphorylated kinetically stable oligomers are added to culture, and you'll see clearly that there is phosphoserine 129 pathology induced. And quantifying this, so again, this was 11 days after addition. These were fixed and imaged. And looking at both survival, so the actual toxicity of these oligomers, as well as quantifying that phosphoserine 129 I displayed on the previous slide, you'll see at certain higher concentrations, the kinetically stable oligomers are toxic. So there's a reduced survival of these neurons, and that's quantified by MAP2 and nuclei and basically cell counting of these at the end of the experiment versus the control. So monomer has no effect on toxicity. These kinetically stable oligomers do. And then quantifying the phosphoserine 129 on the right here, you'll see as the concentration of oligomers increases, you get a greater amount of phosphoserine 129 detected. So in summary of our current Parkinson's disease models, we have alpha-synuclein oligomers and fibrils that we generate in vitro are able to actually induce this Parkinson's phosphoserine 129, as well as aggregation in cellular and rodent models of disease, which is pretty exciting for disease, for studying disease and studying different inhibitors potentially of this process. And we do also have some behavioral changes that we've observed in mice. I don't have time to talk about it today, and it's to be published, but keep an eye out for that. Now looking at Alzheimer's disease. So Alzheimer's disease is characterized by both tau and amyloid beta aggregates. And I'm going to talk about the tau portion of this first. So we have tau normally in cells is going to stabilize microtubules. And again, we have an instance where in solution, it's a very flexible, soluble molecule. And through aggregation, it forms both oligomers and fibrils, and eventually these neurofibrillar tangles that are found in the brain of Alzheimer's disease patients. And we've been able to generate several forms of tau preformed fibrils in vitro. So there are several different splice variants and isoforms and lengths and core of the PHF versus full-length material. And we've been able to generate quite a few of these. For example, the tau K18 fragment, which is induced with heparin. So this is a molecule that helps it fibrilize. We also have the full-length tau that we've expressed in baculovirus, which is hyperphosphorylated, which I'll talk about in a bit, and this smaller tau DGA fragment, so sort of the core of the fibril, which I think is going to be critical for a lot of research going on, as there's quite a bit of evidence now that this is most closely mimicking Alzheimer's disease tau fibrils. So starting with the tau K18, so this is a heparin-induced tau. When this is injected into a transgenic tau mouse, so it has the P301L mutation, we do see it induces pathology. So here we're looking at phosphoserine 202, 205, as opposed to for Parkinson's disease, we're looking at alpha-synuclein and phosphoserine 129. Now we're looking at tau, phosphoserine 202, 205, staining with an antibody. And we see 9 weeks after injection, we do get robust pathology, this 202, 205. Again, this is not phosphorylated going in. It's actually inducing this physiological pathological process of increasing this phosphorylation, which is present in these neurofibrillar tangles. And again, always trying to work towards the most relevant disease model, the thing that most closely mimics human disease. And in human disease, when those tau molecules come off of microtubules, they eventually become hyperphosphorylated, and that's one of the drivers of sort of this oligomerization, fibrillization. And so we wanted to generate hyperphosphorylated tau, and we were able to do this, and it actually fibrillizes on its own without the addition of heparin. So that's removing another variable from the experiment, and hopefully getting closer towards what is really happening in a human brain to recreate in a disease model. So over 30 sites on this tau expressed in baculovirus SF9. So for those of you who don't know, the baculovirus SF9 system is an insect cell system. So it's a eukaryotic system, which allows us to produce proteins with post-translational modifications. And this hyperphosphorylation allows it to fibrillize on its own without the addition of heparin. Then looking at this tau DGAE fragment, so this will actually form fibrils on its own. It's expressed in E. coli, so it doesn't have the hyperphosphorylation. That's something we're also working on. But in our hands, this actually most closely mimics AD isolated PHFs. So there's some literature that's come out recently that's actually looked at the cryo-EM structures of these things and come to a similar conclusion with this DGA fragment that it actually most closely looks like these paired helical filaments that are isolated from actual Alzheimer's disease patient brains. And in our hands here, we have, compared to the AD isolated PHFs against primary mouse hippocampal neurons after 7 days. So these are treated, and then they are fixed with HTTA, which removes all soluble tau species. And then basically the T49 antibody, which is a mouse-specific tau antibody. So this isn't going to recognize this human DGAE fragment we've added. It will only recognize the mouse material that is not soluble, that's deposited. Because of the fixation method, it removes anything that's soluble. And what you'll see here, and that's quantified on the left, on the right here, T49 in yellow, you'll see that there are actually endogenous mouse tau aggregates induced by the addition of these human DGAE PFFs. And you can see it quantified there on the left compared to the Alzheimer's disease isolated material, which also induces a similar effect. And compared to some of the other fibrils and the heparin-induced ones, we didn't see that effect. So we feel like we're on the right track to developing the most disease-like preformed fibril here. Now looking at the other side, so the amyloid beta side of things. So you'll see essentially this amyloid beta peptide is normally in this large transmembrane protein. The peptide ends up being cleaved off, and then it aggregates into oligomers and fibrils and deposits in these amyloid plaques. And we've been able to generate from a synthetic peptide in the lab both oligomers and fibrils of this amyloid beta 1 to 42 peptide. So here's the TEM. From left to right, we have monomers, so there's not much material visible there. Oligomers, these nice round globular structures, and then fibrils on the right, these elongated amyloid fibrils. And here basically showing the same thing on AFM. So we have the monomers, oligomers, and fibrils. And on a Western blot, so SDS gel, Western blotted with the 6E10 antibody, which just sees all amyloid beta essentially. You can see a very distinct signal on a, even after SDS treatment and Western blotting. Monomers run at about 5 kilodaltons, the peptides around 4.5. And then for the oligomers, you'll see a bit of a dimer-trimer signal, as well as the smear for the oligomers at larger molecular weights. And for the fibrils, that's even higher, and we also get a deposit in the actual well of the gel. So a unique signal on a Western blot for these different materials. And that translates into actual toxicity. So again, looking at primary rat cortical neurons, and here we're just looking at toxicity, and you'll see on the left there, compared to control, the monomer and vehicle control, there's no difference here after 11 days or 14 days, sorry. In the middle panel, we have increasing concentrations of our oligomers, same concentrations tested as monomer, and what you'll see is a dose-dependent toxicity of the oligomers, as well as on the right of the fibrils. So they are, due to their unique structure, inducing toxicity, whereas monomer peptide on its own is not having that effect. So there is a unique structure there. And so interestingly, more recently, beyond amyloid beta 1 to 42 peptide, which has had a lot of clinical development, a lot of failed clinical development, there is this pyroglutamate 3 amyloid beta. So essentially what happens here, and you can see a schematic on the right, is the normal A beta 1 to 42 peptide is truncated by proteases, and then it's modified by this glutaminyl cyclase, which basically cuts off the first two amino acids are cut off, and then the third glutamate is cyclized, and this increases its hydrophobicity and neurotoxicity. And it's the presence of this modified amyloid beta peptide in plaques increases as Alzheimer's disease progresses. So while many A beta 1 to 42, the full-length peptide targeting drugs, whether they're targeting the oligomers, the fibrils, have failed, there is still clinical development going on for this sort of more degraded, modified, higher presence in later Alzheimer's disease species of this amyloid beta. And recently, we've been able to generate fibrils, again, from a synthetic peptide. So this pyroglutamate 3, amyloid beta 3 to 42 peptide. Again, on the left, a unique fibril signal compared to monomers. So we get that sort of deposit in the well of the gel. And then here we have, on the right, TEM and AFM images of these nice long fibrils. Interestingly, these do seem to aggregate and form these fibrils more rapidly than the unmodified amyloid beta 1 to 42 in our hands. And no data yet today, but we do have toxicity comparisons underway for both the 1 to 42 versus the pyroglutamate 3 A beta. So in summary of our Alzheimer's disease models, tau fibrils generated in vitro do induce Alzheimer's phosphoserine 202-205 pathology in transgenic mice, although some of these novel heparin-free fibrils may better mimic disease. So the baculovirus expressed full-length tau with the hyperphosphorylation that aggregates on its own, as well as this DGAE fragment that appears to more closely mimic the pathology of an Alzheimer's patient-isolated paired helical filament. And amyloid beta oligomers and fibrils do exhibit dose-dependent toxicity in primary neurons, although some of the clinical failures of that target have led us to develop and pharma to target these pyro A beta 3 to 42 constructs, which we're excited to see what the toxicity looks like for these. So finally, and most interesting, some of the newest stuff we've developed here actually involves cross-seeding. So we've been able to generate these copolymer alpha-synuclein tau fibrils. So these fibrils actually have units of alpha-synuclein and tau within them and will seed each other. So here, and this is immuno-TEM, and what we've labeled are a gold conjugated anti-alpha-synuclein, so 6 nanometers. That's the smaller units, smaller black dots on the image here, and then an anti-tau 12 nanometer conjugated antibody. So those are the larger black circles. And you can see within the same strand, you have both of these individual units. And this has been done before. So this was first demonstrated back in 2003. And literature has suggested that this cross-seeding is occurring possibly within amyloid inclusions. So very interesting to see where this cross-seeding concept goes for Parkinson's and Alzheimer's therapeutic development as we understand these things more and more. So future directions, we want to demonstrate behavioral deficits in mice for both the Parkinson's and Alzheimer's disease models. And we do have for Parkinson's mouse model some behavioral deficits, which will be published hopefully soon. We are currently looking at these recombinant aggregates in microglia. So how are PFFs being taken up by microglia, processed by microglia, and possibly how are they toxic to microglia, or basically what happens after that. So looking beyond just neurons. And then we want to eventually expand this characterization of these different PFFs and oligomers in more sort of human disease representative models, such as human neuronal iPSCs and organoids, so we can better mimic true human disease outside of sort of mouse models or transgenic models. So with that, I'd like to thank everyone at Stressmark and our collaborators and CROs. So they're all listed within this presentation who provided the data for us. They've been integral to a lot of our work. And if you have any additional questions or just want some more information, please visit stressmark.com/PFFs for more information. We also have different things like SOD1 and TTR, and are developing TDP43 PFFs as well. So thank you.